Alright, so regarding the alpha keratin that we studied right now, so there is one thing that you know, that if you do like a permanent waving, that's the girls usually try to do to curl their hairs or like even to straighten the hairs. So there is alpha keratin that's inside the hair that is affected. Right, so we studied alpha keratin, and in the alpha keratin, so as we studied that the hair has alpha keratin and the nails and other things that has alpha keratin, but the hair especially, like if we know that the permanent waving or like curling of hair, so in that alpha keratin is has a major role, or like how it affects. So when the hair is exposed to moist heat, it can be stretched, right? So at the molecular level, when you see the alpha helices, right? So the alpha keratin has the alpha helices. So the alpha helices of the alpha keratin of hair are stretched out until they arrive at a fully extended beta conformation. So what will happen? So the alpha helices will be converted to beta conformation when hair is exposed to the moist heat. And on cooling, when we cool down the hair and cooling, they spontaneously revert to the alpha helix conformation, right? So when it cool down, it will go back to the alpha helix conformation. So the characteristic stretchability of alpha keratins as well as numerous disulfide cross linkage is basis of the permanent waving. So how they do the permanent waving or permanent curling of hair? So the hair that is to be waved or curled is first band around a form of appropriate shape. So like whichever shape they need, like they will first band the hair, and then a solution of reducing agent. So permanent waving or permanent curling, right? So what will be done? Appropriate first appropriate shape will be given to the hair, right? And then a solution of reducing agent, then the solution of a reducing agent, usually a compound containing, so which compound it will be used, like thiol or sulfhydryl. Thiol or sulfhydryl group is then applied with heat, and this is applied with heat, right? And then after that, the reducing agent, what this reducing agent will do? Reducing agent will split the cross linkage, will split the cross linkage, will split the cross linkage of disulfide bond to cysteine residue, right? So first the hair is bent. So what's the procedure? They will bend the hair to appropriate shape. Then will apply the reducing agent, the compound that contains like a thiol or sulfhydryl group, and the sulfhydryl group will split the inside the molecular level. What the sulfhydryl will do? The sulfhydryl will split the cross linkage of the disulfide bond that is in the alpha keratins and will provide to sustained residue. All right. And with this, with the reducing agent, it's applied with the moist heat. So the moist heat, what the moist heat will do? All right, so I will write it down. So moist heat will break the hydrogen bonds. Will break the hydrogen bonds. All right, and right, so this reducing agent will split the disulfide bond and to provide the sustained residue. With the moist heat will break the hydrogen bonds, and this too eventually causes alpha helices to uncoil, right? So the coil that is formed in the structure of polypeptide chain, so the polypeptide chain is to uncoil, alpha helices are polypeptide to uncoil, right? So, and after some time, so there is a rest, and then after some time, the reducing solution is removed, right? So this solution is removed after some time, and an oxidizing agent is added to establish a new disulfide bond. So what will happen? Okay, so this forms. So, so after some time, this reducing agent is removed. Agent is removed, and oxidizing agent is added, right? And oxidizing agent is added to develop new disulfide bonds between cysteine residues of the adjacent polypeptide chains, right? But not but this disulfide bond will be not the same as the as before the treatment. All right. So write it down. So disulfide bond will not the same as before treatment. All right. And then after this, the hair is washed and cooled. So what happens? The polypeptide chain revert to their alpha helix conformation. And after this, hair washed and cooled. And so then what happened? The polypeptide chain will be revert to their alpha helix conformation. To alpha helix. All right. So the hair fibers. So the hair fibers that are treated now curl in a desired fashion because the new disulfide cross linkage. This new disulfide cross linkage will exert some torsion. This new disulfide will exert some torsion or twist on the bundles of alpha helical coils in the hair fibers. Right, so this is the process for the permanent waving that is done. But the hair straightening is not truly permanent because the hair grows in the new hair replacing the old. The new hair that is replacing the old, the alpha keratin has the natural pattern of disulfide bond. Right, so we studied about the alpha keratin. Now we'll study about the second one. So that is collagen. All right. So same like the alpha keratin, collagen also has also evolved to provide strength, and the collagen is found in connective tissues. Right. So what type of connective tissues such as like tendons, examples, right? Tendons, all right, tendons, then cartilage, organized matrix of the bone or the cornea of the eye, right? So these are the examples where the connective tissues are found. And the collagen helix is a unique secondary structure. So for quite distinct from the alpha helix. So for the collagen, what type of structure it is? So it is a left-handed alpha helix. So it is not a right-handed. Do you remember we studied about the left-handed and the right-handed alpha helix? So for the collagen, this is the left-handed alpha helix. So for the collagen, this is the left-handed alpha helix, and for this, it has a quite distinct structure. Like I'll just make a section here. Like for each turn, like for every turn, it has right here. Each turn it has 3 amino acids. Right? It has 3 amino acid residues. residues. Right, so at each turn it has 3 amino acid residues, and which are those? So those are proline, all right, then glycine, and 4-hydroxyproline. So that's the abbreviations for all of this three. So in each turn of this alpha helix for the collagen, so I'll put it alpha helix of this collagen, it has 3 amino acids at each turn. So, but the collagen has a very distinct tertiary and the quaternary structure. So, what happens in that? So, for this collagen, so I will write it here, tertiary and quaternary structure. Right. So, for this tertiary and quaternary structure, so there is three polypeptide chains, three polypeptides that are called alpha chains. So do not confuse the alpha chains with alpha helix. So the alpha chain is just a subunit of a polypeptide, but the alpha helix is the secondary structure of a polypeptide chain. All right. So alpha chain, what is alpha chain? It's just a subunit of a polypeptide, but the alpha helix is the secondary structure of a polypeptide chain. So for this collagen, the tertiary in the quaternary structure, there are three separate polypeptide that is called alpha chains that are super twisted about each other. So these three are super twisted about each other. This super twisting, this super helical, it's also called super helical twisting, is right-handed. Right, the separate chains are left-handed, but the super twisting is right-handed in the collagen, opposite in the sense of the left-handed helix of the alpha chains. All right. So, and for this, you can see the structure on your screen. So this is the tertiary or the quaternary structure of the collagen that you are seeing on your screen. And so there are many types of collagen in the vertebrate, and typically all the collagen contains about so what they contains about 35% of glycine. So all right, this is the content, and 11% of ALA, that's alanine, 21% of proline, and also it contains 4-hydroxyproline. That is this is an uncommon amino acid. Alright, so I will write down full name here. 4-hydroxyproline. Right, so this too has 21%. And this 4-hydroxyproline has a special role in the structure of collagen as well. Right, so the 4-hydroxyproline is really important, and what's the special role? So we'll take a slight turn and we'll talk about the scurvy. Scurvy, why scurvy and what is scurvy? You know, that's a disease that's caused by lack of vitamin C or what's called ascorbic acid, right, or ascorbate. Right, so vitamin C is required for, among other things, so the hydroxylation of proline and lysine in collagen. So why vitamin C is required? Is required for hydroxylation of proline, and this is required for hydroxylation of proline and lysine in collagen. All right. So, and the scurvy, that is a deficiency of vitamin C, right? That deficiency is characterized by general degeneration of connective tissue, and like what it includes, the manifestation is like numerous small hemorrhages caused by fragile blood vessels, tooth loss, poor wound healing, and reopening of old wounds, bone pain, and degenerations, and eventually heart failure also can happen in this vitamin C. And milder cases of vitamin C deficiencies are accompanied by fatigue, irritability, and increased severity of respiratory tract infection. So the most animals make large amounts of vitamin C. So how they make? Like converting glucose to ascorbate in four enzymatic step, right? But in the course of evolution, humans and there are some other animals like gorillas, guinea pigs, and fruit bats, they have lost last enzyme that was needed for the conversion of vitamin C or the conversion of glucose to ascorbate. All right, and humans and these other animals need to obtain the ascorbate from the diet, right? And the vitamin C is available in wide range of fruits and vegetables. Right, so vitamin C, the L, that is also L-ascorbic acid. It's like white odorless crystalline powder, right? So, and that is freely soluble in water and relatively insoluble in organic solvents. So the appropriate intakes of the vitamin C is still in dispute, though. The recommended values in the United States is 90 mg for man, right? Milligrams, like there is some other 75 mg for women. The United Kingdoms recommend 40 milligrams, Australia recommend 45 milligrams, Russia 50 to 100 milligrams. So it still depends, but along with that, citrus fruits and among all other fresh fruits goods. So along with the vitamin C powder, there is like citrus fruits and among all other fresh fruits, it's a good sources of vitamin C. And also there is like peppers, tomatoes, potatoes, and broccoli. So vitamin C of fruits and vegetables is important. So what is the molecular level procedure for vitamin C? What happens and why ascorbate is so necessary for good health? So why it is important? So remember we discussed, right? So this is the amino acid sequence for the collagen. That's glycine, X and Y, where X is a proline and Y is a 4-hydroxyproline. So this 4-hydroxyproline plays an essential role in folding of collagen and in maintaining its structure, right? So this 4-hydroxyproline is important in which role? For folding and maintaining the structure, right? And the proline ring is normally found as a mixture of two puckered conformation, right? So in the structure of collagen there is a proline ring. So this ring look like something like this. Now I will draw just a random diagram. All right, so this is like this here. All right, so this is the amine, and there is this. All right, something like this, and it is something like this. All right, and there is, so this one is C gamma endoproline, right? For example, and there is one another one. So all right, it has the same thing here and the amine group, right? But it differs here. What it differs here, it has, oh, wait. So why it differs here? All right, so it has a hydroxyl, and this amine same as this, for example, and this is CY axo for 4-hydroxyproline, right? So for example, so this is, for example, the proline ring that is in the collagen that is found as a mixture of two puckered conformations. So it has like a two types of conformation, something like this. So that is C gamma endo and C gamma axo. So what happens? The collagen helix structure require what it requires, proline or 4-hydroxyproline in the Y position, right? So this is the amino acid sequence. So the collagen structure will require proline or 4-hydroxyproline in this Y position, right? This is the amino acid sequence, and it will require in this Y position to be in this CY axo conformation, in this conformation. If the collagen needs this conformation, this kind of, it will require proline or 4-hydroxyproline in this Y position. And it is this conformation that is enforced by hydroxyl substituents. So this is the hydroxyl substituents at a C4 in 4-hydroxyproline, right? So this is the hydroxyl substituents, right? So it requires 4-hydroxyproline for to enforce the hydroxyl substitution at this C4. It will require this hydroxyl substituents in the 4-hydroxyproline, right? So the collagen structure also requires proline or 4-hydroxyproline in the exposition as well, right? So it has proline and 4-hydroxyproline. And why? To synthesis or to make this conformation CY endo conformation. And this is the introduction of 4. And for example, if instead of the proline, the introduction of 4-hydroxyproline is in this position, it can destabilizes the helix, right? So if it is here 4-hydroxyproline, so that will destabilize the helix. Why? Because in the absence of vitamin C, cells cannot hydroxylate the proline at Y position. So when there is a lack of vitamin C or absence of vitamin C, cells cannot hydroxylate the proline at Y position. So cells cannot hydroxylate proline at Y position. So on this positions, if there is no vitamin C, cell cannot hydroxylate the proline, and this leads to collagen instability and the connective tissue problems that is seen in scurvy. So this is why that happens, right? So however, the hydroxylation of specific proline residues in the procollagen, the precursor of the collagen, requires action of an enzyme, that is proline 4-hydroxylase. So which enzyme it requires? Proline 4. It requires this enzyme action, and this enzyme is an alpha 2 beta 2 tetramer in all the vertebrate, right? So the proline hydroxylating activity is found in the alpha subunit. So each alpha subunits contain one atom of non-heme ion as well, that is Fe2 plus, and the enzyme is one of a class of hydroxylases that requires alpha ketoglutarate in their reaction. We'll see the normal prolyl 4 hydroxylase reaction. Right, so I'm just drawing the structure here. That is CH2, that is CH2 as well, and this is CH2. This is connected with this and this, and it should connect with this, right? So this is what this is. This is proline residue, right? So when this proline residue reacts, so one molecule of proline residue will react with the alpha ketoglutarate, right? So this requires ketoglutarate as well. So we'll draw alpha ketoglutarate. All right. All right. So this is alpha glutarate. Right, so this is alpha glutarate and plus the oxygen. All right, so this one molecule of alpha ketoglutarate and one of the oxygen binds to the enzyme, right? So it will bind to the enzyme, and this will, it also has the Fe2 plus, right? So in this reaction, the alpha ketoglutarate will be oxidatively decarboxylated to form CO2 and the succinate. So this will give, what it will give to us? As 4-hydroxyproline residue. All right, so this is, and same as here, this is the residue of 4-hydroxyproline. Okay, and that is here CH2. All right, that joins with this C, right? And this is 4-hydroxyproline plus the it will give the succinate and the CO2, COH, that is CH2, CH2 and the COH plus CO2, right? Right, so this is the proline 4-hydroxylase reaction. So in that reaction, proline is when reacted with alpha ketoglutarate, it will give the 4-hydroxyproline, and then the residues are oxidatively decarboxylated to form CO2 and the succinate. So the remaining oxygen atom is then used to hydroxylate an appropriate proline residue in procollagen. All right. So in this reaction, there is no ascorbate needed in this reaction. But however, in the proline 4-hydroxylase also catalyzes an oxidative decarboxylation of an alpha ketoglutarate as well, that is not coupled to proline hydroxylation. So this is it, if it's not coupled to proline hydroxylation. So during that reaction, the heme Fe2 plus that becomes oxidized, and that will inactivate the enzyme and preventing the proline hydroxylation. So the ascorbate consumed in the reaction is needed to restore the enzyme activity by reducing the heme ion. Right. So however, if it's not the prolyl 4-hydroxylase, that also catalyzes an oxidative decarboxylation of alpha ketoglutarate, and that is not a coupled to proline reaction, so it will not be the proline hydroxylation. And during that reaction, the heme, that is the Fe2 plus, becomes oxidized, and if this oxidizes, it will inactivate the enzyme and preventing the proline hydroxylation. So that is why the ascorbate consumed in the reaction is needed to restore the enzyme activity by reducing the heme ion. So what will happen? So with the alpha ketoglutarate, so it will react with the ascorbic acid, right? And then it will reduce the Fe2 plus oxidation. All right. So the tight wrapping of the alpha chains in the collagen triple helix provides tensile strength greater than that of a steel wire of equal cross section, right? And the collagen fibrils. So collagen fibrils, like if it's just collagen, and then if there are too many collagens joining, it will be a collagen fibrils that are supramolecular assemblies consisting of triple helical collagen molecules. Right, you can see the image of collagen fibril on your screen. So the collagen fibrils are supramolecular assemblies consisting of this triple helical collagen molecules associated in a variety of ways to provide different degrees of tensile strength. And the alpha chains of these collagen molecules and the collagen molecules of the fibrils are cross-linked by unusual types of covalent bonds. So what happened in the alpha chains of collagen molecules and the collagen molecules of fibrils. So let's write it here. Alpha chain of collagen molecule and so alpha chain of collagen molecule and the collagen molecules of fibrils are cross-linked by unusual type of covalent bond, right? So these two will have covalent bond involving, and this will involve like lysine or 5-hydroxylysine or histidine residues that are present at few of the and this are present at a few of the X and Y positions, right? So this will have few types of covalent bond. So these are that, and this covalent links create an uncommon amino acid residue that is such as dehydrohydroxylysine non-leucine. So this will have an unusual type of covalent bond that will involve lysines or HYLYS. So that is 5-hydroxylysine or even histidine, right? Resulting, so this can be replaced in some of this X and Y positions, and this links will create an uncommon amino acid, and this links even create an uncommon amino acid residues such as, so that's a really long name. Okay, so it will create dehydro, then hydroxylysine, nor leucine. So this is the name of the uncommon amino acid that is created. So this is the long uncommon amino acid that are created if this is regarding this cross link, and the increasingly rigid and brittle character of connective tissue results from this accumulated covalent cross links in the collagen fibrils. If this collagen fibrils accumulate, it will results in the rigid and brittle character of connective tissue. So a typical mammal has more than 30 structural variants of collagen. So what is a normal variant? A typical mammal has more than 30 structural variants of collagen, and particularly to certain tissues, and each somewhat different in sequence and function. But some human genetics defects in collagen structures illustrate the close relationship between the amino acid sequence and three-dimensional structure in this protein, right? But there are like some genetic defects, and that will really show the close relationship between the amino acid sequence and the three-dimensional structure in the protein. The one is Osteogenesis Imperfecta. All right, so this is characterized, the osteogenesis imperfecta, that's a genetic condition, and that is characterized by abnormal bone formation in babies. And in this, at least 8 variants of this condition with different degree of severity, right, that occur in the human population. So this has almost 8 variants, right? And there is Ehlers-Danlos syndrome as well. Ehlers-Danlos syndrome, that is characterized by loose joints, and it has at least 6 variants that occur in the humans. And in this both disorders, so some variants can be really lethal, and whereas other causes lifelong problems, right? And in all this, in this conditions, all of the variants of both the condition results from the substitutions of an amino acids with the larger R group. So it will substitute, such as like with the uh, cysteine or serine, for a single glycine residue in the alpha chain. So in this two conditions, it will be substituting by this. All right, so okay, right, it will substitute this glycine with either cysteine or serine. Right, in this condition, it can be substituted with the larger R groups in the alpha chain in one or another of the collagen proteins. So different glycine residues in each disorder. So this single residue substitution have a catastrophic effect in the collagen function, because they disrupt this glycine XY sequence that gives a collagen a unique helical structure. Given its role in the collagen triple helix, glycine cannot be replaced by another amino acid residue without substantial deleterious effects on the collagen structure. So this glycine cannot be replaced by another amino acid residue without substantial deleterious effect on collagen structure. Right, beside this, there is one more example for that is silk fibrin. I'll write it down here. I am not doing anything. So we'll just for the knowledge. So silk fibrin. So the fibrin, that's a fibrin, the protein of silk that is produced by like spiders or insects, and its polypeptide chain are predominantly in the beta conformation, and fibrin is rich in alanine and glycine residues, permitting a close packing of beta sheets and interlocking arrangement of R groups. So this was all the alpha helical structure. This fibrin is an example of beta conformations. The overall structure stability by extensive hydrogen bonding between all the peptide linkage in the polypeptides of the each beta sheet, and by optimization of the van der Waals interaction between the sheets. And this silk does not stretch because the beta conformation is already highly extended. However, the structure is flexible because the sheets are held by the numerous weak interaction rather than by covalent bond, such as disulfide bond in the alpha keratin. So that's why this structure is flexible and this structure is not flexible. Alright guys, so that was protein tertiary and quaternary structure. Alright guys, so that was the fibrous proteins that we discussed about, the keratin, the collagen, and the silk fibrin. So next we'll talk about the globular protein. Right, so thank you guys for watching, and don't forget to subscribe the channel. Thank you. We'll see you in the next video.